Yes, so welcome back to Excelsior Academy. So this is the second lecture of plant tissue culture and the techniques. So today we are going to talk about the basic requirements in order to carry out the PTC. So before we go with this, please go and watch lecture 1 so that it becomes very easy for you guys to understand that's what we are going to discuss in the lecture 2. Okay, so please keep on watching likewise, in the order so that the concepts will be clear to you guys. Now, the first thing what we required in order to carry out the plant tissue culture is, of course, the plant material. Okay, so this should be disease free and should not be old. Now, why? Because the totipotency is not going to work if we are using any uh, old plant material, and it has to be disease free. So, if you have listened the first video, then you know that how we can get the disease free tissue, that is from the meristematic tissue. It will be free from the viruses and bacteria. Now, why they are free from these bacteria and viruses? Because they are fast growing cells, and that's why it is free of these viruses and bacteria. Also, the particular species and variety genotype which are used should be of the right one. Okay, you just can't go into the garden or into the forest and get some random sample of leaves. You need to know what plant material you want to go for the plant tissue culture. So that is required. Generally, in vitro germinated seedlings are frequently chosen, as seed is often also much more readily sterilized than the softer plant tissues. Now, when plants are healthy and at the desired stage for use, it is often the case that only a specific part of these plants will give the best explants. So, इसका मतलब क्या हो रहा है? See, if you have got the entire plant is there, okay? So, it's not necessary that each of these parts will be acting as the explant. They can act as the explant. But the thing is that it depends on the condition or the totipotency of that particular plant sample, which will give you the better results. You can take the roots, you can take stem, you can take shoots or leaves or flowers, seeds, stamens. You can take any part and you can carry out the tissue culture, that is plant tissue culture. But among all these, which one will be the best will give you, it will be determined only after carrying out these experiments. So basically we go with the meristematic tissues, okay, and mainly the young part of the leaves, or that is the particularly the internode. Now, equipments and the glassware. So what kind of equipments and the glasswares are required? So, of course, the incubating chamber or the laminar airflow cabinet, which has got the UV light. Okay, and now all these are required for the aseptic transfer, because first of all, we are taking the disease-free part from the plant, and after that, we need to maintain that aseptic conditions, for which we need to have all these laminar airflow cabinet and UV light is there. So when we use this laminar airflow, we have got the 3 switches over here. You can see that. So one is for the UV, one is for the tube light, and one is for the fan. Okay, so before you start, you put your UV light at least for 10 to 15 minutes on prior for your any experiments. Then after that, you switch off the UV, then switch on the fan, and then the tube light. Okay, then you carry out, you raise a shutter, you carry out all your experiments, wipe the entire thing with the alcohol. So basically we use 70% alcohol. Okay, for the sterilization of the surfaces, and after that you carry out your experiment, you transfer it aseptically. Everything has to be done under the sterile condition only. Now once your work is done, close the shutter, then close your fan, switch off your fan and tube light, and then again switch on the UV for another 10 to 15 minutes. Okay, so this is the procedure by which you're going to use the laminar airflow. Now you can have laminar airflow either vertical or horizontal. Now, what does it mean? Vertical means like this kind of airflow will be there. Like from the top, the air will be coming. Horizontal means from the this side where it will be coming towards you. Okay, so basically, we work with the horizontal only. Okay, moving on to the next. Then we have got the incubator. So this incubator will have the temperature control of course, present 5.5 degrees Celsius. And the recommended temperature of tissue culture is around 36 degrees Celsius. So depending on the type of the plant, you can have the temperature. For example, if you're growing a cold resistant plant, like which you can see in the, top of the mountains or which are growing on the snowy area. So that time you need to keep the temperature low. Okay. Then autoclaves. Autoclave is required for sterilization of the glasswares and the media. So that is like, if you remember, that is the temperature is around 121 degrees Celsius. 121 degrees Celsius. And for 15 minutes, we keep the sample at this particular temperature. And after that, it is cool off. And again, sterilize aseptically, it is transferred to the laminar flow wherever you're carrying out your plant tissue culture. Okay. Next is refrigerators and freezers, that is for the storage of reagents, tissue culture, stock solutions, chemicals, etc. And hot air oven, dry sterilization of glasses and media. Yeah. So all these equipments will be present wherever the tissue culture experiments are carried out. Okay, so microscope, this is needed in order to, to understand if suppose any contamination is there or not. In order to check all that, we need then pH meter to adjust the pH of the media, washing of the equipments. So basically, we wash all the glassware and the pipettes. If they are made up of glass, so that needs to be washed every time you use it. Okay, and if suppose it is the plastic, then you cannot reuse it, so you have to throw. So those are those ones which we just use and throw ones are there. But if it is glassware, then you need to wash it, then again sterilize it in the autoclave, and then dry it in the hot air oven. Yeah, it's a long process actually, but yes, all these are required in order to carry out. If suppose the pipettes are made up of these polypropylene things, then you don't have to wash it. You can actually throw also those which are made up of the plastic ones. Okay. Then water purified, because every time you're going to use the deionized water, it is going to be free of all the bacteria and the contaminants. So whatever the water you get, you need to again sterilize it in the autoclave, and then only you use it for the culture. Centrifuge, in order to increase the concentration of the cell suspension culture. Shakers, so in order to provide the aeration to the media, so that the plantlets or the cells can grow in the suspension culture. Balance, in order to to weigh all the nutrients which is required to prepare for the media. Then shells, so that you can keep it, of course. So each member will be having their own shells. Scissors, scalpels, and forceps, this is in order to you are going to cut. So how exactly we do is, even if you can bring your sample into a plastic, after that what you do is, in the petri dish you keep this plant. Okay. So suppose, let's say that you have taken the leaf only. So let us say that you have got the leaf. Then with the help of these scissors and scalpels, what you do is, you actually first you sterilize it. Okay. Surface sterilization, what you do is called surface sterilization. And after that, you take all these scalpels and forceps, scissors, you make a small tissue. Okay. Then this tissue again are cut into small sections. Then each sections are taken and put into the test tubes carrying the media. Then they are put into the, if it is a cell suspension, then they are put into the shaker. If it is a solid media, then it is directly put into the incubator. Okay. Okay, then culture vessels. So again, they are made up of borosil glass. So obviously they are preferred ones. So this also includes test tubes, conical glass bottles, special flat tubes, etc. So that depending on the what kind of experiments you're carrying out. So initially what we do is, whenever we have got the small explants are there, initially we start with the boiling test tubes. Once that has been done, and if you get a good amount of experiments, sorry, if you get a good amount of results, then it is transferred to the bottles, and then it is transferred to the greenhouse chamber. Then the glasswares like measuring cylinders, beakers, funnels, petri dishes, this is, I mean, no need to say, because these are all present in the labs. Then miscellaneous, you need to use the non-absorbent cotton. You cannot use the cotton absorbent, because that is going to absorb all the moisture and all everything. So aluminum foil is also required, because it can be used to cover the exposed part of the plug. So it will become wet during the autoclave. So you can just cover it so that it doesn't get wet, because if it gets wet, then there are chances that it will have the bacteria containing in that, which will actually get spread during a tissue culture experiments. Then normal pen marking, everything is needed. And each researcher should have their own markers so that they can label it whenever it is required. Then microwave, it's not essential, but sometimes what happens is you are actually making your media, agar solid media you are making, right? But unfortunately, you are getting late and you leave the lab, and next day you are coming. So again, you're not going to sterilize the media, because continuous sterilization will also, degrade the nutrients which are present in the media. So what you can do is just melt the media using the microwave, or if you have hot water bath, you can put it over there also. So just melt the media and again use it for the pouring into the petri dishes. Okay, so that's all about the basic requirements which is needed for the plant tissue culture. So keep watching, there are more to come, different types of cultures method also. So thank you guys for listening. If you like it, please share and subscribe with your friend circle also. If any doubts are there in the upcoming videos also, you can ask me in the comment section. Thank you.